University, Solapur. He obtained his BSc degree from Gulbarga University in 1991 and MCA degree in 1995. He has completed his PhD in computer science in the year 2007 from Gulbarga University. The title of his thesis is A Study on Pattern Classification and Analysis of Medical Images. His research interests are character recognition, document image analysis, neural information processing, fuzzy theory, deformable models, image processing, pattern recognition, data and image mining. He has worked at various universities and institutes such as Government Boys College Raichur from 1997 to 2007, Government First Grade College Vijayanagar Bangalore during 2008, Karnataka University Dharwad from 2008 to 2012, and currently he is working as Associate Professor and Head Department of Computer Applications at Solapur University. He has guided 2 PhDs and 16 MPhil candidates and guiding 8 PhD candidates. He has published 35 papers in international and national journals and 45 papers in international national conference proceedings. He is the author of 2 books, namely Medical Image Analysis and Multilingual Document Image Analysis. He has got 12 years of research experience and 17 years of teaching experience at graduate and postgraduate levels. He has organized 5 conferences and workshops. He has delivered invited talk in various conferences and seminars. He received grants from UGC and AICTE, and he has visited Hong Kong, Australia, USA to present his research papers in international conferences. Such a great personality is with us today. On behalf of organizing committee, I once again welcome him and request him to share his knowledge. I request him with a bouquet.
Can you hear me? Okay. So I am very happy that the organizers invited me to deliver a talk. They considered me as one of the resource persons, and I am very thankful to the principal of the institution, and especially my special thanks to Dr. Hegadi. And just I want to mention that my association with Hegadi is very long back. We were classmates in from 8th to PUC second year in Gulbarga, and after PUC second year he switched over to engineering. I moved to BSc, MCA, and we didn't had a contact. And once in 2008 when I was working in Karnataka University Dharwad, I attended a workshop at IIT Kharagpur, and it was organized by Biomedical Engineering Department at IIT Kharagpur. And there was one resource person called Dr. Manjunath, and just I was interacting with him, and he told that he was from Karnataka itself. He worked in this Bapuji Engineering College, and he mentioned the name of Hegadi sir, that he was colleague of him. So that's how I came to know that Dr. Hegadi is working here, and then later we contacted, and now we have association. In different ways we are associated with each other. So today I asked what kind of information, what kind of material I should deliver. He told little bit focus on the research. I think most of you are the aspirants for PhD. Am I correct? Okay. So I will little bit focus on how you can initiate, because there is a great need for good PhD students. Everyone want to do PhD, but none of them want to do work. So this is the very bad principle that is being adopted by the students. And we are in search of good students. We get many students, many people approach us, but getting a good student is very difficult. So I will give you a little bit hint which can definitely help you in achieving getting a PhD seat and doing your PhD very successful. First thing is you focus on what topic you want to work. That should be very clear. You should be very clear on which area you want to work, on which topic you want to work. So there should be a clear focus on topic, and then you must have a deep knowledge in that area. When you focus on that area, you must have a deep knowledge. So getting deep knowledge is through reading a lot. You have to do a lot of survey, you have to do study, you have to refer good papers. Again here many journals are there. There are plenty of journals. Always prefer good journals, good conference proceedings, something like IEEE, ACM, Springer, elsewhere. Don't go for paid journals or even survey work, because most of the content of the paid journals will be copied contents. And second thing is, facts are not reviewed. If you give a paper, within 1 week they will give the result, and most of the results will be accepted unless your paper is really bad for rejection. So that's why the content of the such journals is highly unauthenticated. So don't go for referring such journals. So we didn't had during our whatever I am going to present today, it is my PhD work, and that was I think almost 10 years old. So technology has changed a lot, but still with the intention that it will give you some idea how you can start your work and what approach you can take up during your PhD work. I am going to present this. So if you want to really do join for a PhD, you have to be focused on the topic. You have to be very well knowledge in the area, and definitely you will get guides. There is no dearth of guides, but only guides will prefer good students. So you have to show that, you have to perform, you have to show that you have ability to do work. So with this few words, let us start my discussion on the medical image analysis. Basically, I worked on endoscopic images, even though topic is medical images. My focus was endoscopic image. So I just want to tell you how I started. When I joined for PhD, our guides, none of our department guides were knowing what is computer science PhD, because one was from mathematics and another one was from statistics. So it was our task. We ourselves has to find the problem. We have to ourselves identify. We have to ourselves start working on that. So it happened such that I used to develop small software when I was working in Raichur, and I had a contact with a doctor where he was in need of a software for his endoscopy device. So I delivered some 2-3 software, and one among them was, his requirement was endoscopy. And that's how I had a very easy access to endoscopy images. And the doctor was very conversant, very friendly with me. He not only gave the images, he also gave me the information about the images. What kind of images, abnormal, normal, all those details. And even he helped me a lot during the manual segmentation, because without the physician's knowledge, without the physician's help, we cannot do any medical image. We are not experts in medical images. We have to take the help of them, right? So that's how I visited many institutions. I started searching for the topic, and finally I thought, why don't I should do some work in endoscopy image, because I was visiting that hospital. I came to know the kind of problems that were existing in endoscopic images, and my focus, of course we had many problems in the endoscopic images itself, ulcer and other kind of things. I focused on the cancer tumor regions, and I started working on that. So my initial work started with a very elementary problems, and then we moved on, and we have done many works in that area. I will briefly present. I will focus on the kind of techniques we have used and what kind of results we got, because of the time constraint. I cannot completely present. It has basically more than 90 slides. I don't think I will be able to discuss everything on all these slides, but I will focus on the kind of work I have done. I will try to show you the results. So when we look at the medical image, basically the computerized image processing and analysis of medical image cover large number of potential topics. So when we look at, you can just see every medical equipment need a software, a hardware and a software. Okay, so doctor cannot do all the decisions. As just now we had a session on Dr. Ravindra. He was telling what was the technology 15 years back. and what is the current technology. So how it is happened? How it all happened? It is because of the instrumentation development, hardware development as well as software development. So these two combinations have brought the today's technology. You take up any equipment, any medical equipment, it must have a technology, a hardware technology as well as a software built on that. So basically any medical image processing tool or technique will have image acquisition, and it will have image formation and reconstruction, then image enhancement, image compression and storage, because medical images are very huge, large in size, and we don't want to compromise on the quality of the image. So the compression and storage are very essential component of the image, medical image processing. Then image analyzing. So once we capture the image, once we store them, once we enhance them, then the next stage will be to analyze the content of the image, and then we have to come out with the decision, that is image based visualization. So these areas led to the development. So based on these areas, many development happened. Like for example, every image processing tool require image segmentation. So you need to understand what is image segmentation, how we perform these segmentations. There are plenty of techniques. Then image matching and registration. Particularly for some images, like almost most of the medical images require the image matching techniques and image registration techniques. Then motion tracking and change detection from image sequences. So particularly these are very essential in understanding, analyzing the heartbeat and all, where the heartbeat, maybe the motion of the blood flow, all these. When we want to analyze such kind of things, then motion tracking and change detection techniques are very much essential. Then measuring of anatomical and physiological parameters from the images. So these are also a part of the image processing techniques. So the research work related to these methodologies are crucial for obtaining solutions for problems like. So based on these, the techniques what just now we have discussed. So it led to the image guided surgery and intervention. Nowadays surgery, most of the surgeries are carried out with the help of the image processing tool as well as software. Okay, so earlier entire surgery were carried out entirely by physicians based on their understanding, their idea, but nowadays most of the techniques are most of the medical imaging techniques are being used for surgery as well as intervention. Atlas based description of entire anatomical regions, deformation analysis based on biomechanical and other models. So deformation analysis, this is very helpful, particularly to understand the moving components of the human body. Next, visualization of anatomical and physiological processes. So these are the things generally we do with the help of the medical images. These are the motivating factors for researchers in the past 2 decades. So if we look at the development of the medical image analysis, the medical image analysis started somewhere around 1970s or 80s. It is not as old as that is computerized medical image analysis. It is not much older one. Since as we know, during 1950s the computer innovation happened, and with the innovation, the initial usage of the computers were in the particularly image processing, were in the area of the satellite communication. And later people started thinking, why don't these computers and imaging techniques can be used in the medical also. So if we look back where the image processing were used, it can be divided into 4 areas, 4 stages. So the research on medical images from last, it is more than 30-35 years. It can be divided into 4 areas, that is between 1980 and 84, it was an era of 2D, 2-dimensional images. At that time 3D was not in the focus. So people used to analyze the 2-dimensional images using the computers. And later from 1985 and between 1985 and 1991, so people started thinking on developing the knowledge on the image, just not looking at image itself. They stored the knowledge of the image, and they stored they used this knowledge for analysis purpose. And later we find between 92 and 98, the full 3D image analysis and mathematical model driven approaches were in full swing. People started developing 3-dimensional visualization of the images. That's how we got MRI, CT scan, all these technologies. And 1999 onwards, there was a great revolution that happened in the imaging, medical imaging. More advances happened in the use of computer technology for medical images. And later we can even find that the image-guided procedures, image-guided surgery and all were carried out with the realistic visualization of the images. So let us focus on the topic on which I have carried out my research. It was endoscopy. As all of you know, most of you know, endoscopy is a long scope which contains at the tip of it, there are many equipment such as light source will be there. There will be surgical devices and even water tubes will be available which can suck and which can flush the water in the human body. So there are different types of scopes basically. My focus is on endoscopy in which the tube is passed through the mouth and it will go into the human body. It passes through the stomach, esophagus, stomach. Okay, then it enters into different parts. It may not go all along till the anus, but most of the parts can be visualized with this endoscopy. So endoscopy is fitted with a device, and this device shows the inner picture, motion picture basically. And if you have connected to a computer, you can capture the video, you can capture the clips of the inner components. And in the later development, even many analysis tools have been developed for understanding the inner part of the human body. So it's a endoscopy means, as I told you, it's a looking inside, and medically it means looking inside the human body, and it's a minimal invasive diagnostic. We need not do any surgery to perform endoscopy. So it's a minimal invasive diagnostic procedure, and with this many of the it helps us in extracting the biopsy, and this biopsy can be used for analyzing the kind of content, whether it's a normal tissue or it contains any kind of abnormality such as the cancer growth and all. So endoscopy can be used to diagnose or treat many disorders such as the inflammatory bowel diseases, peptic ulcers, then many such kind of diseases can be analyzed with the endoscopy. A physician may also discover an apparent abnormality. So basically the tumor growth and all can be monitored. A patient can be subjected for endoscopy repeatedly, and they can watch whether any tumor found inside, it's a really a abnormal growth, or such as it has a cancer region and all. So this kind of growths can be analyzed with the help of endoscopy. So my first technique was a very simple technique we used with we started our research with a very simple technique, what we call it as the color image information based segmentation, in which we chosen like normal region, abnormal. Under the abnormal we chosen the initial cancer region and the advanced cancer region. So based on the content of the pixel information, we chosen a particular category of pixels as the normal pixels. Then we chosen a particular category as the initial stage cancer region, and then we chosen a particular region of pixels as the advanced cancer region. And based on that, I will just show you. So first we have acquired the image, then we have pre-processed. Basically in the pre-processing we applied some smoothing technique and all. Then we have extracted the features. So as I told you, based on the color content, the knowledge what we had, based on the color content, empirically we selected this color content, and based on the color content, we classified the image as a normal region, the region with advanced cancer or initial cancer stage, and then if any bright spots will be there or lumen region will be there, that also we eliminated, and finally we classified the image regions as normal and abnormal. So I will show you, these are the values that we have selected for that is for red component, green component, blue component. As we know, image contains three components: RGB, color image. Endoscopy is a very good color image. So we selected a red component, green component, blue component, and based on that initial stage and advanced stage, these are the values. And we computed the mean and standard deviation. And in the statistics, we have that most of the 95% of the values lie between the 3 sigma interval. Okay, when we draw the standard deviation, most of the values lie between 3 sigma regions. So based on that, we selected the values and we classified the images as normal image with the image with the initial stage of cancer and images with the advanced stage of cancer. So this is the processing stage. We have taken the image and we applied a smoothing in the second, and then we extracted the initial stage cancer region and advanced stage cancer region. And these are the results what we can find. These are some of the abnormal images and these are the results of our proposed method, and we validated our result with the help of the manual segmentation that was being carried out by the physician. And the accuracy, it was almost, we didn't analyze on the accuracy at that time. You can just imagine it is a 10-year-old work. Okay, so but we found that most of our segmentation technique was in coincidence with the whatever the segmentation is being carried out by the physician. In another work, I took up one paper from one researcher from Hong Kong, and he worked on the curvature analysis. As we can see, the endoscopy, when a cancer growth has happened in the endoscopy image texture will entirely differ. So we computed the curvature of, we first extracted the edges and then we computed the curvature. So you can see here I have put up the 2 images. One is the abnormal image, another one is the normal image. If we obtain the edges for the normal image, the edges will be very, very smooth. Whereas the same for abnormal image, the edges will be very rough. Okay, the curvature of the edges will be highly varying. So we extracted this curvature property and we combined this with the RGB segmentation. So I'll just show you the result of this. So first we performed the RGB segmentation as just now I explained. Then we computed the curvature for the edges obtained after the RGB segmentation. So I'll just directly go on to the results. You can find this is the original image and this is the after RGB segmentation. We performed canny edge detection technique. There is one edge detection technique. How many of you know image processing? Okay. See, I am coming across many of the terms which are image processing terms. So if you know it, then you will understand better. Otherwise, I think it is a bit difficult because I cannot start explaining what is image processing. Okay. Anyway, wherever it needs explanation, I will focus on that. So there are many edge detection techniques. So one such edge detection technique is called as canny edge detection technique. So we extracted the edges from the image and we selected the larger edges. Okay, many edges are obtained there. So a larger edges are selected because smaller edges may be due to noise. So we eliminated the smaller edges, we analyzed the larger edges. So one such edge is selected here and we applied. This edge is, generally there will be there will be too much variation in the curvature. So once again we smooth this curvature and we obtain the curvature value. This is the plot of its curvature value, and again we smooth this curvature profile. You can find the curvature profile for an abnormal image. It's highly varying. Okay, there is a zero crossing. So we count how many zero crossing happened. So zero crossing means when the line crosses this red line. When this curvature crosses the red line, we will count how many curvature crossing happened, and based on that we can decide whether the image is normal or abnormal. Here you can see we have, basically we compared our result with the one which was developed for curvature computation, and it was done by one author called as Krishnan and others. So this is the work done by them, and these are the images, the results of our proposed method. You can see the result of the Krishnan method. We cannot find a larger curvature profile, whereas in our method a large curvature profile can be obtained. So that's how our method is an improved one as compared to the method proposed by Krishnan. Again, what we did, we compared our result with the abnormal and normal images. You can see the normal images curvature profile is very, very low. First of all we get very less number of edges in that. And whereas in the abnormal images we get more number of edges and larger size edges also. And these are the curvature profile of the abnormal images, and these are the curvature profile of the normal images. So this is how we classified. There is another technique which has been used for classification of normal images and abnormal images. One more simple technique we have, that is the initially we have used the RGB segmentation. Now we have used HSI segmentation, hue, saturation and intensity segmentation. So the color image can be modeled in the different ways. One, we have RGB, that is the natural model, and then we have hue, saturation, intensity. We can describe the image, color image in that way also. Then we have the CMYK model. There are many models, image color image models. We have selected the HSI model because in some medical equipments, HSI model is also preferred rather than using the color that is RGB model. So with that intention, we choose an HSI model, and this is the method or algorithm which has been used for HSI model. Initially we took the color image, we converted that image into HSI image, and then we separated the hue component, saturation component and intensity component, three components. So then on the intensity component we obtained the complementation, that is the negative of the intensity component. Then we perform thresholding. Thresholding basically converts any image into two regions, that is black and white, okay, binarize. It basically binarizes the images, and parallelly for the saturation component also we applied thresholding. Then we found out the product of hue component and this binary thresholded image, and then finally we merged these two images. So whatever we got, that is the HSI segmented image. Okay, the final result of that we got as a HSI segmented image. So these are the stages of the whole image processing technique. You can see this is the normal image, this is an abnormal endoscopic image, and these are the different stages what I have just now discussed. Okay, so finally we obtain the last image, that is this is the image. Okay. Yeah, this is the HSI segmented image, and we have actually this is the HSI segmented image. We have obtained the edges for that, and then we have extracted a larger edge from that, and we have drawn a curvature profile on this. So this is how we refined our work by adopting different techniques. Then we also, you can see the result for the normal images and the abnormal images. Here also it is very evident that this technique also is very successful in segregating the normal and abnormal images based on the curvature profile. So then we used one more technique called as watershed segmentation. This is a very well-known technique. This is one of the morphological image processing technique, and we have used this, and we combined this technique with the RGB and HSI segmentation based techniques. So okay, so before moving on to that, let us understand what is this watershed segmentation technique. As I told you, it is one of the morphological techniques. Basically, with the watershed segmentation technique, ultimately we are going to segment the image. We are going to obtain the image basically it divides the image into regions based on the regional properties. There are basically the image segmentation can be classified into two main categories. One is energy based approach, another one is watershed segmentation based approach. I will come to the energy based approach, where we are going to study about the active contours, which are very helpful for particularly medical images. But water segmentation technique is also a very basic technique, even though it is not that much effective, but that also we have used. Okay, so let us come to my results on this. This is the algorithm I have proposed. So we have used around 50 images, normal images, and 50 abnormal images, and basically this watershed segmentation technique divides the image into number of regions. If more abnormality is there, more number of regions can be obtained. So we set up, based on the experimentation, we came out with the result that 30 regions, if there are more than 30 regions, the image can be treated as an abnormal image, and if the number of regions obtained are less than 30, then it can be treated as a normal image. So you can see here, we have a very abnormal image, and these are the different stages of the watershed segmentation based techniques. And finally we obtain a watershed segmentation region, and this is the labeled region where we have different color for different regions that we have obtained. And you can see this is an abnormal region. We have obtained a very large number of regions due to this, and you can see here there are many abnormal images, and these are the regions, number of regions that have been obtained for these abnormal images. So we have compared our results with the normal images also. When we perform same technique over the normal image, the number of regions obtained were very less as compared to the abnormal images, and that's so I think we have put the threshold as a 10, not the 30, and you can see all the normal regions have less than 10 regions as compared to the abnormal images. So these are just analysis in the form of a graph. All the red points are shown are related to the abnormal images, and all the blue pixels or points are related to the normal images. So now later we combine this with the RGB segmentation. So without doing any segmentation, we as it is applied the watershed segmentation technique, and later we combine this with the RGB segmentation. I will directly move on to the results. See, all these works are available in the form of paper, okay, and they are all downloadable, right? So anyone who want to refer my work, you can directly put my name, and all the papers are downloadable. They are available in ResearchGate, and they are available in academia.edu. Okay, those who are interested, you can just download these papers directly from these websites. I have kept all papers open, right? Okay, so you can just see with the inclusion of RGB based technique, you can see more number of regions are obtained here, okay, in the segmentation technique, and it can still better enhance, increase the number of regions as compared to the previous technique. So these are some of the abnormal images and their results, and you can see the number of regions are very large as compared to the previous technique. They are almost more than, some of them are near to 100. So very great number of regions are obtained here, and we have compared this with the normal images. So our accuracy is still increased as compared to normal and abnormal images using this technique. Later we applied same thing. We tested with the HSI segmentation and combined this with the watershed segmentation, and let us directly move on to the results. So this is the kind of results what we got with the help of the HSI segmentation and then in combination with the watershed segmentation technique. So yeah, again these are the results of some of the abnormal images. So 5 abnormal images. So here also performance is not as great as RGB segmentation, but still we have large number of regions obtained for abnormal images, and very few regions are obtained for the normal images. So the proposed segmentation technique had accuracy, again as I told you, I had a standard 50 abnormal and 50 normal images. So the proposed method could perform 96% of accuracy it has achieved for abnormal images, and 92% of accuracy for normal images. So these are all the watershed segmentation based techniques. Later I came out with the active contour techniques. So first, before moving on to the results what I got or the techniques what I used, it will be helpful if I focus on the active contours. What is the active contour, and basically why it is used? The active contour is something like, suppose I have an object and I want to extract the or I want to segment the object. I have to just initialize a region over the object. There are many active contour techniques, but generalized techniques I am going to explain. It uses two kinds of energies. One is the internal energy, another is called as a external energy. It can be treated something like a balloon. Okay, as we push the air into the balloon will start increasing in its size. Okay, so two types of, one is the external energy, that is external force will keep the balloon round shape. Okay, balloon is always round shape. And second thing is internal energy, what the air we are pushing, that will help in expanding the balloon. If you take a rubber band and put the rubber band over some object, okay, larger object, then what happens? The rubber band will take a minimum shape. Okay, why it takes a minimum shape? Because again in the rubber band two energies will be acting. One is the energy which will pull it towards the center. Okay, so that's how it will occupy the minimum area. Another one is the area that is the object itself, that will not allow beyond its boundary. Okay, so this rubber band technique also can be used for explaining the active contours. So in the active contours, we try to minimize these two energies. Basically, we wherever we acquire, when external energy is equal to internal energy, then wherever the curve take the shape, that will be the boundary of the object. Okay. So based on this technology, the active contours will work. Active contours are deformable. It has many names. Even they are called as the snakes. Okay, deformable models. Many names are there for active contours, and there are variety types of active contours. We will just focus on some of them. So just by changing the parameters, just by changing the way of approach, we can go for different types of active contours. So active contour are perhaps the most successful class of techniques used for medical image segmentation. See, active contours are very helpful in medical image segmentation. It is because the medical images are there is a smooth variation in the image information. That is one thing in medical image. You take up any medical image, the variation in the information is very smooth. There is no rapid variation. There is no rapid variation found in the medical images as we can find in the natural kind of image. The object, whenever there is an interface between one object and another object, we can find a clear separation of objects. Okay, so that is the difference between medical images and normal other general types of images. So when we take up the medical image, active contours are very helpful because these active contour techniques are very very adaptable to the smooth variation in the image. They will they will try to segment, they will try to identify boundary when there is a smooth variation in the image. So active contours or snakes can be used to segment objects automatically. These are automatized techniques. Once we need to just initialize the boundary, okay, just we have to show the object and then it starts deformation. Deformation means it starts either contracting or expanding, and it will try to occupy. And again there are, as I told you, varying methods. In some active contour, only one curve initialization and only one region will be extracted, and many other active contour techniques will break themselves. Okay, will break themselves and they will occupy different regions based on, suppose we are segmenting different regions in the image, they will break themselves and they will occupy different regions. So the basic idea is the evolution of curve or curves subject to constraint from input data. As I told you, we will start with one curve. Okay, the curve will start evolving. Okay, and it may be a single curve evolution, or it may be curve itself can divide and it can start evolving in different regions. So the deformable models work based on deformation of geometric structures as well as propagation of the curves under the influence of internal and external forces have been considered. So there are basically, to understand active contour, we must have the knowledge of physics also. Why? Because we have to understand how force works. So basically two types of forces are acting here. One is the internal force within the object that will try to pull the curve towards the center, and another one is the external force, that is the image force, whole entire image force. So that's why the understanding of the physics force knowledge is very important here. An attempt to classify these techniques can lead to the following distinct categories. So as I told you, there are different categories of deformation, the active contours. So some of them are classified under these heads, but there are actually more number of classifications. So one is called as deformable templates. So this class of approach is the least flexible one, since it relies on models that are made up of low-level geometric components such as lines, circles and ellipses. So this technique requires one initialization. We need to initialize one curve or one closed curve, and it will deform, and it's not a flexible technique. Why? Because the technique used in this, the basic techniques used in this particular deformable templates are such as lines, circles and ellipses. So it is less flexible technique, but it doesn't divide itself. It ultimately come out with only single region. And second one is point distribution model. In the point distribution model, basically we need to specify the points. We need not give the initial region. We need to just specify the points where the abnormal region is available, and it takes up the shape and it starts deformation. So a step further in the rigid and articulated models was the use of specific patterns of the variability from a representative training set. So instead of entirely giving the region, here we need to mention the points where abnormal region is present, and that's why it is little bit improved one, but not as improved as the next techniques. So one more set of techniques, that is snake and active contour models. The snake model is a major breakthrough in computer vision and made use of the curve propagation approaches quite efficiently. See how snake moves. You can see snake moves like everyone knows. Okay. So snake is very flexible in its movement. Okay, it's very flexible, and this flexible technique was used in this active contour snakes. And this is a very major breakthrough. Earlier we only used certain properties such as lines, curves and ellipses, but this technique helped really a lot, particularly in medical image segmentation, and presently people are using this technique. And next we have another category of technique, that is geometric flow model. That is one more technique, one more category. Here a novel class of techniques based on the propagation of planar curves to use with the various applications like boundary extraction, segmentation, stereo tracking, etc. So this is another set of techniques, what we call it as geometric flow. Then the active contours without edges is implemented. So we have used the active contour models or snakes. Under that again we have two categories. One is with edges and without edges. So basically the medical images doesn't contain sharp edges. So that's why we used active contours without edges technique in our implementation. So the technique of active contour without edges as proposed by, there is one classic paper called by the author Chan and Bese. So these people developed this active contour without edges technique. We implemented their technique in different ways. So we initially selected the input color image and we converted that into grayscale image, and we used the energy minimization function. As I told you, two types of energy acts in the active contours. One is the internal energy, another one is the external energy. So our task is to minimize this. That means internal energy must be equal to external energy. When the deformation starts, initially internal energy and external energy will differ. So it will try to bring the curve in the region where this internal energy and external energy will be equal or same or minimum. Okay, we cannot achieve lesser than this. So the energy minimization, for that we used a algorithm called as a Jacobi method. So again, as I told you, this paper is available. If you want to see more about it, you can just download. Initially a seed point is selected, so we need to initialize a curve. So we have initialized a curve, and the initialization should be done in such a way that, suppose we have a tumor in the image, we have to initialize nearby tumor. If you are initialized somewhere else, it will not bring the curve near to tumor unless the tumor is very strong and there are no other objects or artifacts in the image. Suppose you have some bright spot in some other region, the curve will pull towards that. So you have to make sure that the initialization has to be done across the tumor, around the tumor or near the tumor. So the initial, right, the circle is so obtained will be initial contour which deforms in each iteration. So basically this active contour technique is computationally extensive. It requires large number of computations. You will see later we have analyzed all these techniques and we have seen how each and every technique, how computationally expensive based on the number of the storage space, the time taken, all these analysis we have done. So it's a computationally extensive, but the accuracy of the result is very good as compared to any other techniques. So in the meantime, the curve may divide itself. So this technique is a self-division. It divides itself. Suppose for example we have multiple tumor regions or multiple abnormal regions in an image, it will divide itself and it will multiple regions are extracted from the abnormal images. The process finally generates region in the image depending on the image texture, and the region boundaries will be the region minima. So that's how it extracts the regions where the boundary of that region will be minimum. So again, as I told you, many parameters we said, okay, these all things are not so relevant. I will just move on to the results. You can see these are some of the abnormal images, their grayscale images, and after applying the active controls, you can see very clearly